Thank you, and good morning, everyone. We're going to start off today by talking about what fit-for-purpose clinical trial quality means and how it's achieved. Also, I want to focus my presentation on the importance of clinical trial quality through the lens of the clinical investigator and the sponsor investigator. So today, we'll discuss FDA's perspective on clinical trial quality and data quality that is fit for purpose. We'll talk about how quality by design and risk proportionality principles are applied in the design and conduct of clinical trials. We'll discuss implementation of risk mitigation strategies and proactive measures at the site level to avoid protocol noncompliance and errors that matter. And then I'll provide a case example that illustrates the benefits of adopting quality by design and risk proportionality approaches to the design and conduct of clinical trials, both from the clinical investigator and the sponsor investigator perspectives. So how does FDA define clinical trial quality? Well, unfortunately, you won't see a definition for clinical trial quality defined in our IND regulations under 312. So what I have on this slide is the closest that I can point you to in our regulations that specifically discusses quality in a clinical trial. And that's under 21 CFR 314.126, which broadly describes what constitutes an adequate and well-controlled study. So what's on this slide is the primary basis for determination of whether substantial evidence of effectiveness has been provided to support the labeling claim. FDA does review protocols submitted under an IND prospectively to assure that the study is adequate to permit evaluation of the drug's effectiveness and safety. So we can also turn to guidance to better understand clinical trial quality. ICH E8 Revision 1 or R1 is an international guideline that was issued in 2021 and discusses general considerations for clinical trials. This guideline describes internationally accepted principles and practices in the design and conduct of clinical studies that will ensure the protection of study participants and help with the acceptance of data and results by regulatory authorities. The guideline provides recommendations on how to build quality in the design and conduct of clinical studies across the product life cycle, and this approach is also known as quality by design. It helps us to understand quality in the context of how trials are conducted. And the guideline states that clinical trial quality is considered as fitness for purpose, which means that the quality and amount of information generated from the clinical trial should be sufficient to support good decision making. I also want to acknowledge the contributions of the Clinical Trials Transformation Initiative, or CITC, in this quality by design space, as they have been working on this quality by design approach for over 15 years. CITC is a multi-stakeholder public-private partnership that was founded by FDA and Duke University in 2007. To help us further understand this idea of quality as fit for purpose, CITC also defined quality in clinical trials as the avoidance of errors that matter to decision making. Specifically, we're talking about the errors that could affect the safety of trial participants or the credibility of trial results. So this is another way to help us understand quality in clinical trials. So if we delve into FDA regulations under 312 and our guidances, it is clear that multiple parties play an important role in trial quality. So it's the sponsor who designs and oversees the trial and analyzes and reports results. It's the contract research organization to whom the sponsor has transferred their responsibilities to. It is other service providers that are contracted by the sponsor and CRO to perform critical clinical trial activities. It's the IRBs who provide initial and continuing review and approval of research. And of course, It's the clinical investigators. So those of you who translate a trial from the pages of the protocol into the clinic, you have a very important role in delivering quality. And it is your day-to-day work engaging with participants and carrying out protocol requirements and collecting trial data that are the foundation of trials that yield quality data and reliable study results. So now let's go back to ICH E8R1 to further explain this fit-for-purpose clinical trial quality, which is rooted in the foundational principle of quality by design. And quality by design entails proactively designing quality into the design and conduct of clinical trials to ensure the protection of the rights, safety, and well-being of study participants, the generation of reliable and meaningful results, and the management of risk to the study's critical to quality factors using a risk proportionate approach. And this last bullet encompasses a lot. And I'm going to talk about this in detail throughout my presentation today. And I really, really hope it's not to the point of ad nauseum. But at its core, this last bullet really entails identifying the critical aspects of a trial, specifically the critical data and processes, and directing efforts towards these factors. This approach ensures that we effectively address what is essential for generating credible and reliable data while protecting participant safety. So initiating the QBD process, or the quality by design process, during the design phase of the trial is crucial for its success. And again, this is another point that I'm going to underscore throughout my presentation, because I believe this is where the process often falters or goes wrong. So first, why is this done at the design of the trial? Well, the goal of a clinical trial is to generate reliable information to address research questions. Clinical trials are vital for drug development, and a well-designed and executed clinical trial is not only essential for regulatory decision-making, but also for evidence-based healthcare decisions. Conversely, a poorly designed trial or an ambiguous protocol can lead to a poorly conducted trial and introduce significant issues such as compromising participant safety, producing inadequate or unreliable evidence that could impact decision-making, wasting resources and the time and efforts of investigators and participants, and it may not align with ethical standards. So to further illustrate this point, imagine starting with what appears to be a perfectly constructed house. However, if the foundation and structure aren't built properly, issues begin to surface as you start using the house. Similarly, in clinical trials, if quality is not built in from the start, issues can arise during the study. And we really want to avoid that firefighting approach where you're scrambling, making hasty decisions, applying quick fixes without thorough investigation, and addressing symptoms rather than root causes. What we want to prevent is a study plagued with data integrity and reliability issues, a study that might look something like this house. And this could lead to rejection of the data or even the entire study, and it could jeopardize regulatory approval and even result in the discontinuation of potentially beneficial therapies. So the protocol serves as the blueprint for ensuring quality. So for those sponsor investigators in the crowd who are designing trials during protocol development, you really have the opportunity to de-risk the protocol and avoid risks to the data and participant safety. So design is really the point where you can identify those critical aspects of the trial and take steps by tailoring the design to avoid the kind of errors that impact regulatory decision making. And it gives you the opportunity to engage with key stakeholders to get their input. Protocol design and development is also an opportunity to ask, why have we included this? Whether it be, data collected or some procedure being conducted. and it gives us the opportunity to streamline the trial where feasible to reduce the burden on sites and other study personnel. It's also that last checkpoint to verify that the trial you've designed will answer that important scientific question you had in mind when setting out to design the trial. And it gives you the opportunity to highlight important risks that are not eliminated through study design and that may be mitigated through the trial's operational plans and procedures. So we say that you should avoid errors that matter. And what errors matter? Well, they are the errors that impact your study's critical to quality factors. So what are these critical to quality factors? Well, ICH E8R1 defines them as attributes of a study whose integrity is fundamental to the protection of study participants, the reliability and interpretability of the study results, and the decisions made based on the study results. So we're talking about critical data, critical design elements, and critical study processes and procedures. If their integrity were to be undermined by errors in design or conduct, then the reliability or ethics of decision-making or the results of the study would also be undermined. So really, in simple terms, the question we need to ask ourselves is, what do we really need to get right to ensure the reliability of results and patient safety? So for those sponsor investigators in the crowd, you should conduct risk assessments to identify and review the critical to quality factors during the design and planning of your study, and then re-review and reassess them throughout study conduct, analysis, and reporting. So these critical to quality factors are individualized to your study and study design, and inflexible one-size-fits-all approaches should be avoided. So simply identifying all studies to always have the same broad categories of critical to quality factors should be avoided. The critical to quality factors should be communicated to all those involved in the trial conduct to ensure a shared understanding and alignment on what is critical to quality and the quality expectations. So let's move on to challenge question 1. So which of the following best defines the concept of quality by design in clinical trials? A reactive approach to quality control that addresses issues after they occur. B, a proactive approach that emphasizes designing quality into the trial from the outset. C, a method focused solely on data collection and statistical analysis. Or D, an approach that prioritizes regulatory compliance over participant safety. So I think this is an easy one. The answer is B, a proactive approach that emphasizes designing quality into the trial from the outset. So the next challenge question, 2, in implementing quality by design, which of the following elements is most critical for ensuring clinical trial quality? A, randomization of participants. B, a comprehensive understanding of the study's critical to quality factors and potential risks to those critical to quality factors. C, strict adherence to standard operating procedures. Or D, extensive documentation of all trial activities. And I think the obvious one, again, and an easy one is B, comprehensive understanding of the study's critical to quality factors and potential risks to those critical to quality factors. Okay, so let me pivot now to ICH E6R3, which is a draft guideline, and it's an international guideline for good clinical practice. So you'll hear more about this updated guideline, revision 3 in particular, from Dr. Ayalou this morning in his presentation that follows mine. And this guideline has been totally reorganized and renovated to include 11 principles and 2 annexes. So Annex 1 provides recommendations on how the 11 principles can be appropriately applied to clinical trials. And Annex 2 provides additional considerations on how these principles may be applied to clinical trials that incorporate various operational approaches, such as trials that use decentralized or pragmatic elements and use real-world data. So the purpose of the E6R3 update is to provide recommendations that can be applied across the broad range of clinical trial designs and settings. These updates are intended to address the complexities of clinical trials in the current global regulatory climate and to facilitate modernization, harmonization, and innovation in clinical trial design and conduct. So the guideline is grounded in these concepts of quality by design and risk proportionality, and it supports fit-for-purpose clinical trial quality. Adopting these approaches, so quality by design and risk proportionality, is what allows flexibility in applying the E6R3 principles and recommendations. And this requires critical thinking to tailor solutions such that they are fit for purpose for your clinical trial. So the appropriate measures are taken based on the specific risks and needs of the study to ensure the overall study results and participant safety. So here are the 3 foundational principles of the 11 that are in ICH E6R3. And these 3 in particular support this risk proportionality approach. So at a high level, Principle 6 focuses on quality by design, highlighting that quality should be integrated in both the scientific and operational aspects of clinical trial design and conduct. Principle 7 stresses that trial processes should be implemented in a way that's proportionate to the risk to participants and to the importance of the data collected. And then there's Principle 8, which advocates for simplifying protocols to ensure operational feasibility and to reduce burden on sites. And so it's all of these principles together that help to minimize unnecessary burden on participants, on sponsors, and on investigators, and to ensure that resources are allocated effectively and efficiently by focusing on what matters most to quality, again, to participant safety, and to yielding reliable results. So the overall message that I'm trying to convey is that quality by design is the essential ingredient to managing trial conduct proportionately. Your study plans and processes should all be guided by your quality by design approach, your identified critical to quality factors, and your risk assessments. Your risk assessments and your management processes should be iterative and adjusted when new risks emerge. And errors to critical trial data or participant safety should be addressed as early as possible through risk-based monitoring, or ideally prevented altogether by building quality into the design of your trial. Much of what I presented on today occurs during the trial design phase and really falls on sponsors and those sponsor investigators in the crowd to design a clinical trial that is scientifically valid, ethically responsible, and focused on producing reliable data that can be used for regulatory decisions. For those clinical investigators in the audience, though, you might be asking, what's expected of me as a clinical investigator, and how do I apply the principle of risk proportionality to my responsibilities? Well, first, it's crucial that you and your clinical trial staff understand what's critical to quality for a study and the potential risks to those critical to quality factors. You should focus resources and investigator oversight on those critical data points and high-risk areas, so data and processes deemed critical to quality, and ensure that the data is credible and reliable and that the critical processes are conducted according to the protocol. You should assess risks throughout the study, adapting practices as necessary to respond to new information or unforeseen challenges that may arise in study conduct. So now I'm going to present a case example on the use of electronic diaries in a clinical trial to illustrate how applying risk proportionality to the study design and conduct can help to focus resources on what's critical to quality and to improve data quality. So this is a hypothetical case, but unfortunately many parts of this case have been taken from actual cases. So two pivotal trials were submitted to FDA to support approval of an investigational drug as adjunctive therapy in patients with a seizure disorder. The two trials were similar in design and were multi-center, randomized, double-blind, placebo-controlled studies. The primary efficacy endpoint in both studies was the change in the mean convulsive seizure frequency during the treatment period compared to the baseline period. The primary efficacy endpoint was an observer-reported outcome that measuring seizure frequency, as I indicated just now. Both protocols pre-specified and described the use of eDiaries by the caregivers to directly enter daily seizures. And the intent was for the parent or caregiver of the study participant to contemporaneously document their seizure, the daily seizures into the eDiary. And the sponsor contracted with a service provider to provide caregivers with the eDiaries and to manage the eDiary data. So this slide illustrates the eDiary data flow, and I'm just going to walk you through the diagram to provide a basic understanding of this study conduct. So each day, the caregivers recorded the daily seizures of the subject, including the dosing of the study drug and the use of rescue medications into the eDiary. And the eDiary data were transmitted electronically from the devices to the service provider's database. If the eDiary device was connected to a Wi-Fi or a mobile data signal, then the diary data was automatically sent at the end of completion of a diary entry. If the eDiary did not have Wi-Fi or a mobile data signal, then the diary data was stored temporarily on the device until Wi-Fi or a mobile data signal was available. So investigators had read-only access to the eDiary database, and they couldn't enter or make changes to the eDiary data. The service provider was responsible for reviewing and managing the eDiary in the database, and they could make backdoor changes. And these backdoor changes to the data were tracked in the ECOA database audit trails. So, for example, if the service provider in the review of the data identified discrepancies, errors, or missing data points, they would generate a data query and would send the query to the site via an online portal. If the query resulted in a change to the eDiary data, then the investigator had to authorize the data change and did this by completing a data change form in the online portal. The service provider then retrieved and reviewed the change form, and once they had all the information they needed, the service provider would change the data in the system. And again, this was all tracked via the system audit trails. So once all the data changes were made and the data were reconciled and cleaned, then the service provider transferred that clean data file to the sponsor for incorporation in the SDTM datasets and for analysis. So during the CI inspections, the inspectors reviewed the audit trails of the eDiary data that were retained at the site. They observed that a portion of each participant's eDiary data was entered by the service provider as much as 1 year after the date that the seizure data should have been contemporaneously entered by the caregiver, so as much as 1 year after the date that the seizure occurred. So why was this done? Well, towards the end of the trial, the sponsor noticed that there was more than an expected amount of missing data and asked the clinical investigators to retrospectively collect this data to mitigate the amount of missing data. So the clinical investigators and the study staff did this by conducting in-person and phone interviews with the caregivers. The clinical investigators also collected the caregivers' personal diaries and notes. So these personal diaries and notes were not designed for use in the trial, but the caregivers typically kept these contemporaneously personal diaries and notes as standard of care for these patients. So first, let me just say up front, when I talk about these inspection observations, that we found almost little to no issues with the data quality for the data that was entered directly into the devices by the caregivers. And then for this data that was retrospectively collected, we just didn't, there was so much of it that we just didn't want to throw it all out. So in order to determine the reliability of the seizure data that was entered retrospectively in the ECOA database by the service providers, we had to look more closely at the source, which were these paper diaries, the caregiver's personal paper diaries, and also other paper records at the site. So it was the progress notes and medical records where this clinical investigators documented their interviews with the caregivers. So these source records were not consistently maintained or retained at the site. So there were a lot of missing source records to support the seizure frequency data that was submitted to FDA. In addition, multiple discrepancies were identified between the source records that were available and the seizure frequency data reported to FDA. So the root cause for the discrepancies in the source data the source records that were available was multifactorial. So first, the first cause was that the caregiver's personal diaries did not collect all the protocol required information. So they didn't collect the time and type of seizures, they didn't document the dosing, or they didn't document the use of rescue medications. So it was really difficult to verify, the information that the service provider entered into the ECOA database, because there was just no source available to support all of the information that was entered. The second cause for the discrepancies stemmed from the service provider staff who was responsible for entering the retrospectively collected data in the database. So they misinterpreted the data change forms that were completed by the clinical investigators, along with the copies of the caregivers' personal diaries that were sent to them, and they frequently consolidated multiple seizure events that occurred on a day into a single seizure event. So, there was a large volume of data that needed to be entered by the service provider, and this, simply was not a planned part of the study. And the service provider staff, really was not adequately trained or qualified to accurately interpret and input this data. So what was the cause of the missing data? So let's go back and talk about that. Well, there were eDiary device design, malfunction, connectivity, and transmission issues that were experienced during the conduct of the trial that were not adequately and promptly addressed by the service provider or the sponsor. There was poor caregiver compliance of the eDiaries for many reasons. There were no contingency plans in place for device malfunctions and connectivity and transmission issues. So, for example, they could have had a backup device when the devices malfunctioned, or they could have had a procedure in place to enter data in the web-based portal for those malfunctions, or they could have used a telephone or IVRS system when there were connectivity and transmission issues. So I think our data reliability assessment and its implications are pretty obvious. But we determined that the clinical investigator had retrospectively collected a portion of the eDiary data for 96% of the randomized subjects in the two studies. Again, this collection occurred up to a year after the seizures had occurred. And, I honestly can't remember what I had for breakfast, let alone what I ate a year ago. So, there is the potential for recall bias here. And so this affected approximately 20% of the seizure diary for the two studies. And because of the potential for recall bias in this retrospectively collected seizure data, this data was considered unreliable, and so a post hoc sensitivity analysis was performed by both the applicant and the FDA on the primary efficacy endpoint with respect to this retrospectively collected data. So what were the lessons learned? Well, first, sponsors or sponsor investigators should conduct risk assessments at the study's outset to identify critical to quality factors and the risks of the critical to quality factors. Sponsors and clinical investigators should prioritize resources and implement targeted strategies for high-risk areas to maintain data quality. So for this case, focus should have been placed on the seizure frequency data, in addition to the many critical processes involved in its collection. So for example, the sponsor could have placed more focus on the design of the eDiary devices or validation of these devices. It could have placed more focus on help desk support, developing contingency plans, and conducting centralized monitoring of the eDiary data to identify this missing data sooner. And for the investigators, they could have placed focus on caregiver training on the proper use of the eDiaries, monitoring for missing data on their end, and perhaps better reporting to the sponsor of issues the caregivers were having with the use of the devices. Sponsor and investigator oversight should be adjusted based on the assessed risk. And because this was critical data, for example, more frequent reviews of this data by both parties could have helped address some of the data quality issues as they arose in the trial. The sponsor and clinical investigators should communicate quality expectations to ensure there's a shared understanding among all team members. And then both parties should foster open communication about potential issues for quick responses and adjustments to data collection practices. So this is one more bonus question because I really want to drive this point home. How should the principle of risk proportionality influence the allocation of resources and the implementation of risk mitigation strategies? A, all processes should be standardized regardless of the level of risk involved. B, risk mitigation strategies should be minimal as extensive measures may hinder the trial's progress. C, resources should be concentrated on high-risk areas and critical data points with appropriate risk assessments guiding decisions on acceptable levels of risk. Or D, the importance of data should be assessed only after the trial is completed based on the outcomes. So of course, the answer is C. Resources should be concentrated on high-risk areas and critical data points. And then finally, my closing thought that I'd like to leave you with, and that is achieving fit-for-purpose data quality largely hinges on the foundational principles of quality by design and focusing on what matters to participant safety and the reliability of the overall study results. Thank you so much. It's been a pleasure to present at this clinical investigator training course, and I'm happy to take questions during the panel session.